Hello everybody. My college name is Ram Niranjan Jhunjhunwala College, and today we are performing the differential count. My name is Aniket Jaiswal and I am from TYBSC Zoology and my roll number is 666. The aim of the practical is to determine the differential count of WBC in the given blood sample. The requirement of this experiment is blood sample, clean slides, distilled water, Leishman stains and microscope and etc. The principle of this experiment is Leishman stains contain methylene blue and eosinophils in alcoholic medium. The eosine stains the cytoplasm while methylene blue stain the nucleus of the cell. The morphological of the nuclei in the WBC helps to differentiate the various types. The WBC are of two types, agranulocytes and granulocytes. The agranulocytes are of three types, neutrophilic, basophil and eosinophil. The agranulocytes are of two types, which is lymphocytes and monocytes. The principle of the experiment after the procedure. In the procedure, take a clean and dry slide and place a drop of blood at the edge of the slide and place the spreader slide at the angle of 45. Pull the slide back and it along the edges. Push the blood forward to the end of the slide with the smooth movement. Dry this blood smear at the room temperature. Strain the smear with Leishman stain for about 5 to 10 minutes. Wash the stain using distilled water to remove the excess stain. Dry the slide in air and observe under the high power of compound microscope. Place the slide on the microscope and focus at the one end of the slide. Move the slides from left to right in a zigzag manner. Count the WBCs and record them in the observation table. Count 100 leukocytes and obtain percentage of each. In them, neutrophils are the size of 10 to 12 micrometer. The cytoplasm stain in a nuclear diagram. The nucleus is multi-lobe, 2 to 7 lobes are there. The granules are very fine rather than basophils. The cells appear pale violet in color. It is comprises 60 to 70% of WBC. The absolute count of the cell is 3000 to 6,000 cell per cubic millimeter. It is phagocytic in function. Next one is basophil. Basophil shows the cell of the size is 8 to 10 micrometer. The cytoplasm stained with basic dye. The nucleus is slightly lobed. The granules are larger in size. The cells appear blue in color. It comprises 0.1% of WBC. The absolute count of this cell is 0 to 100 per cubic millimeter. Next one is eosinophil. Eosinophils are the size of 10 to 12 micron. The cytoplasm has a coarse granule. The nucleus is bilobed and the cells appear red with eosins. The absolute count of the cell is 150 to 400 cell per cubic meter. Next one is lymphocytes. In lymphocytes, the size of the cell is 7 to 12 micrometer. The cytoplasm is basophils. The nucleus is large and round and occurs in major part of the cell. Granules are absent. It comprises 20 to 25% of the total WBC. The absolute count of this cell is 1500 to 2700 cubic millimeter. The function of the leukocyte is for immunity by antibody production. Next one is monocytes. In the monocyte size of the cell is about 16 to 18 micrometer. The cytoplasmic basophilic. The nucleus is round or oval. When the cells are young and become kidney shaped, it's become older. It comprises 5 to 10% of the total WBC. The absolute count of this cell is about 350 to 800 cells per cubic millimeter. The cells are phagocytic in nature. In a well-stained, specific fine radius, blue granules are observed. The observation table of the normal differential count. The cell type is neutrophilic is around 50 to 70% and eosinophil is 1 to 3 and basophils are seen 0 to 1 and lymphocyte is around 25 to 35 percentage and last one is monocytes which is seen 4 to 6%